Hello everyone and welcome back to the Food Microbiology series. I am Tommy and today we're going to enter the last chapter of this playlist. Today we're going to talk about fermented foods. So how we use the microorganisms to create new products, to make new flavors. So let's begin. Okay, if we start, we have the intro. Microorganisms are used for creating fermented food with or without microorganisms. Okay, so we use microorganisms to create fermented foods. And those fermented foods can have microorganisms or not. For example, the bread is a fermented food without microorganisms. But, for example, a cheese is a fermented food with microorganisms. The fermentation is used because it improves the taste, it improves the quality of the food, it also preserves the food, but it also adds nutrients to it because the bacteria add new components that they were not there previously. Then the fermentation also has a detox action on the anti-nutrients of the food, especially in veggies. And with fermentation, we need less time to cook our food. Types of fermentation, we have the alcoholic, the lactic, the acetic or alkaline fermentation. We're going to view them all in this video. The fermentation is affected by the temperature, the pH, the inhibitors that maybe are present in the food composition as well, and the precursors, as well as the concentrations of oxygen and CO2. The system of operations in the industry and the mixing also affect the fermentation. So, because all of these parameters affect the fermentation, the industry uses starter cultures. Starter cultures that can be natural, mixed, or from defined regions. Those starter cultures do contain probiotics. Probiotics is a living organism that improve the host's health because they increase the epithelial barrier and the union to the mucosa. They block the pathogens, they produce the antimicrobial substances, and improve the immune system. So probiotics are living microorganisms that are present in food that will grow in our gut and give us those benefits. If we talk about probiotics, we need to talk about prebiotics. Prebiotics are non-digestible component that is food for the microbiota. The microbiota is the one we have in our gut, in our colon. These prebiotics are, as I said, non-digestible components of our diet. For example, the fructooligosaccharides and the galactose oligosaccharides, as well as inulin, this is soluble fiber, the resistant starches, and they help us with the microbiota growth because it's food for the microbiota. And they improve the immune system as well, because if they grow, then the microorganisms will improve the immune system. If we start, we have fermented meats. We use the fermentation in meats to preserve and to add the aromas. The little cups that are in meat are fermented. The problems that can occur are either the no fermentation, because there are very little to no carbs in certain meats, or the inconsistent fermentation. Okay, so here we have two parts, the microbiota and the impact on the meat. Let's start with the microbiota, and in the ham, we have the micrococci and the staphylococcus, as well as high amounts of mold and yeasts. The staphylococcus is mainly on the surface of the ham, and of course we use starter cultures. In the case of sausages, we use the lactic acid bacteria such as Lactobacillus sakei and Staphylococci are as well used, but they are even inside the sausages, whereas in the ham, the Staphylococcus was on the surface. And for both of them, we use starter cultures. For the reason I explained here, there are very high amounts of variables that affect the fermentation. So that's why we use starter cultures. The impact on the meat, well, the fermentation has proteases that create the sweet and the umami taste. The lactic acid bacteria create the acid, the bacteriocins and the peptidases. The Staphylococci has the nitroreductase. This is for creating nitrate from nitrate. And they have also the catalase and the amino acids degradation. The superficial microbiota, they have lipases, proteases, and mycotoxins. So the flavors are created by proteases and lipases, and the others by lipases, lipooxidases, and volatile amino acids. Then we have the fermented fish. Fish has higher pH than meats, and the deepest fishes of the sea have a lot of urea and trimethylamide. They are sources of nitrogen. The common way to ferment the fish is done by Asians, and it's the union of fish with cooked rice or maize, sucrose, salt and garlic. And the fermentation is at 24 degrees in Japan, for example, or 30 degrees Celsius in Thailand. There are many classifications of fermented fish. We have the solid, the semi-solid, and the liquid fermentation, and also classification based on the final look, the methods used, the substrate, which fish we use, and the quantity of salt as well. Then we have the European fermented fish. In Iceland, we have the hakkarl, which is shark, fermented with lactic acid bacteria. In Norway, we have the rackfish, which is salmon fermented with Lactobacillus sakii. And in Sweden, we have the surströmming, which is harang fermented with Halanaerobium. Then we have the fish sauce with the Tetragenococcus and the Narezushi, which is the typical fermentation. These last two are not really European. And the typical fermentation is the one I described here. So fish smell a lot because of the urea and the trimethylamide, especially the trimethylamide. Having a lot of nitrogen, the degradation of those nitrogen components create a lot of amines, especially in the trimethylamide. It creates volatile amines that they do smell. Then we have fermented dairy. Well, this is probably the most common one. When we use the lactic acid bacteria for the fermentation of the milk, the sugars will suffer either a homolactic or a heterolactic fermentation, or even a heterofermentation. The proteins will suffer the proteolysis to create aromas and flavors, and the lipids, they will create organoleptic properties such as aromas and flavors and changes of the taste mainly. And there is also the production of bacteriocins, bacteriophage resistance, and the autolysis, which is the degradation of the food, of the milk in this case. The types of fermentation we can have is, the first one is the mesophilic, to create the buttermilk, and we have to be careful with the aroma acid balance. Then we have the thermophilic to create the yogurt. This is very common and popular. The bacteria grow very fast. They do the lactose fermentation. pH is around 5, and there are anaerobic conditions. The therapeutic fermentation is for the acidified milk, and it's basically with sterilized milk sterilized, such as UHT, and we added the Lactobacillus acidophilus. And then we have the fermentation with yeasts. The fermentation with yeast creates the kefir. The kefir is becoming popular recently, and the kefir grains have a lot of microorganism variety. We talk about 25, 30, 40 even different types of microorganisms inside the kefir grains. And the original kefir grains have nothing to do with the market, with the industry kefir grains we get on the supermarket. That's nothing to do with the Mongolian original one. Because of the yeast, they create ethanol. They suffer the alcoholic fermentation, and they also create polysaccharides. With malts, we have the fermentation with malts to create the villi, and this is a high viscosity, texture, and it has characteristic flavor. We use the Geotrichium candidum and the lactic acid bacteria as well. And then we have the cheese production. Cheese production and high varieties because of different microorganisms. So if you heard that, for example, France or Italy or even Spain have a lot of cheese varieties, like in France there are millions of varieties, that's because of different microorganisms and different cultures, as well as different concentrations of those microorganisms in those cultures. So the phases we need to follow to create the cheese is, well, the selection of the milk first. Yes, because we can start the fermentation with cow milk, goat milk, or another animal, or even a species of a certain animal, because it has maybe some more fats or other has more lactose. So we have to select the milk we want for the cheese we want to make. Then we have the selection of the starter culture. This is very important. And then milk to cheese. We have the acidification, which can be done by 4 methods. Then the coagulation using the rennet. And then the aromas and flavors. To reach aromas and flavors, we do the maturation of the cheese. Continuing, we have the fermented drinks, such as wine, beer, and vinegar. Starting with wine, we need starter cultures with Saccharomyces ellipsoideus. And wine will suffer the alcoholic fermentation, of course. If you ever heard about the bread aroma, that's because of the Brettanomyces bruxellensis. Then, after the fermentation, the alcoholic fermentation is done, the wines, the Crianza wines, will suffer the malolactic fermentation, and that's done by the Enococcus oeni. And this microorganism will do the decarboxylation of some carbohydrates, some components. It will lower the acidity and it will raise the pH, and this will give more stability to the wine. That's why the Crianza wines that are matured for 3 years, 5 years are very high quality, also because of the stability they have. And then we have the beer. The beer, well, five phases, 5 stages of creation of the beer. The malting of the barley is the first one, then the mashing, then we have to boil the wort and add the hops, and then we have the fermentation with Saccharomyces cerevisiae in the case of an ale, or Saccharomyces uvarvum for the lager. So remember, those 2 types of yeast are very important for the alcoholic fermentation of the beer. And then we have the treatment of post-fermentation, which can be adding more CO2, removing some alcohol, and creating new products from that. And then the vinegar. The vinegar is interesting because it has two ways to be created, with aerobic part and completely anaerobic. So when it has the aerobic part, that means that it has suffered the alcoholic fermentation first and then the acetic one. So for example, in a wine with Saccharomyces ellipsoideus, we get the wine, and then from the wine it can suffer the acetic fermentation and create vinegar. And the completely anaerobic fermentation to create the vinegar is done by the Clostridium thermaceticum, that converts the carbs present in the wine, for example, directly to the acetic acid. And we have three methods for creating vinegar. The Orleans method, which is the let alone, the spontaneous wine acetic fermentation, that's what I talked about. Then we have the drip method, which is the fast German method. And we have the vine branches with acetic bacteria on them. And then we put the wine on top of those branches, and it makes contact with the acetic bacteria, this one, the Clostridium thermaceticum, and as it drops, it will drop as vinegar directly. Then we have the Frings actuator, which is a tank with an agitator, and this is done for the industrial method. This is indeed the one which is used right now. And then as last one, we have the fermented vegetables. The grains fermentation is the most common fermented product on the market. Yes, why? Because everyone eats the bread, and the bread is a fermented product from grains. Bread, it can have either long or short alcoholic fermentation. Have you ever heard of bread long fermentation? Long fermentation bread? Yes. That's because it can have either the long or the short. It's cooked in the oven, so that's why we don't have the ethanol. But this is an alcoholic fermentation, so ethanol is created indeed. And if we eat the bread uncooked, which is not likely to be done, we will have alcohol in the bread. Then we have the fermented veggies, such as the sauerkraut and the olives and the chimichurri, and they are all done with the lactic acid bacteria. Then we have the products from the soy fermentation, the tempeh, the miso, and the soy sauce, as well as the natto and the pidan. Those two are the only ones that are alkaline fermentations. Then we also have the fermented coffee, which can be wet or dry, and the cocoa fermentation, which is done directly below the leaves after the recollection. Okay, so that's it for the fermented foods. I hope you liked the video. Click the like button if you did. Subscribe to the channel if you're new. Share this video to your friends and family to let them know how fermented foods are created by the industry, what are the benefits of eating those fermented foods, as well as what's the difference between prebiotics and probiotics. Remember to download this diagram. I will leave down in the description. It's on my website. You can download it for free. Just ask for it. I will send it to you. Thank you so much for watching this whole series of videos, and see you on the next one. Bye. Hope you enjoyed.